Okay. So my name is Michelle Lacasmo, and this is my presentation on nuclear transport. So this is a figure from the book looking at transport through the nuclear pore complex. You can see here, the cytosolic carboprotein with the NLS is recognized by the importon and connects. Then with number 2, the importon NLX or NLS complex is transported into the nucleus by transporter at the center of the nuclear pore complex. In the nucleus, the importon molecule associates with a GTP binding protein called RAN, and this interaction causes the NLS containing protein to be released for use into the nucleus. Then you can see number 4, RAN GDP importon complex is transported back through the nuclear pore into the cytosol, and the importon is released in the cytosol for reuse. And yes, so that is what this figure is. Okay, so looking at the questions for the different experiments and the figures. So the objective of figure A is the effect of importon beta 1 on ERBB2 in the cytoplasm and nucleus. The conclusion from the bottom panel is that importons bind and help transport things into the nucleus. There's less transport of ERBB2 into the nucleus, and the nuclear fraction with siRNA shows decreased levels of ERBB2 in nucleus, and the cytoplasm is normal. You can see here different changes in the bars. Okay, figure 2, wild type. The objective is wild type and mutant ERBB2 binding with importon beta 1 with NLS. So conclude from the data that the wild type has the NLS sequence that binds to importon beta 1, which you can see there's these 2 bars are present, and then that the mutant lacks the NLS. Okay, so then this one looks at RAN. So the different questions, the role, what is the role of the GTP hydrolysis in transport of ERBB2? So ERBB2 is released when the RAN GDP binds to the importon beta 1. This complex is involved in transport from the cytosol to the nucleus. And how is the transport of this impacted by RAN Q69L compared to wild type RAN? So this mutant type cannot move into the nucleus and remains in the cytosol, which you can see here. There's nothing present here, right? So instead of using this RAN Q69L, could they use RAN GPPNP? Well, that type would, the ERBB2 would not be able to move to the cytosol and would remain in the nucleus. And then the results, how would the results vary? If either of these non-wild types were used, there wouldn't be like free flow between the cytosol and nucleus, and transport would stop. Okay, and then last thing, so transport mechanism of a specific protein that utilizes NLS. So this was in the book. This is SV40 large T antigen. It was used to study NLS, and what they looked at is when one of the 7 amino acids, specifically looking at lysine, is mutated to threonine, the nuclear import of the antigen was blocked. They theorized that this shows that the sequence is necessary for nuclear import, and that NLS is sufficient transport of cytosolic protein into the nucleus. That is it. Thank you.